Hello everybody. First of all, I would like to thank the organizer for inviting me and giving an opportunity to present my work. Um, my talk will focus on the emerging field of network medicine, which provides a platform not only to exploit the molecular complexity of diseases, but for example, identifying the disease genes, pathways, or the disease modules, but it also provides us to explore more about the disease relationships. So here you can think about a disease as a perturbation of network. And what we have in the last decade with the human genome sequencing project, we have got a list of gene and gene products, and we have collected them, but if you see them individually, they are not so informative to reveal much about the complexity of biological systems. And moreover, being at the Human Genetic Conference, the central goal of human genetics is to identify the genetic risk factors for complex diseases, Mendelian disease, and that's what we all are doing. And there are different technologies, like starting from candidate gene linkage studies, genome-wide, and now we are focusing on whole genome sequencing. But we all are now aware that that's it's not about identifying the genetic risk factor, but when you move from genotype to phenotype relationships, there is a long biological path, and it has many intermediates, which can be microRNA, which can be the interactions between the different components, which are altered by these genetic changes. So what we believe and what we strongly believe is that the network can provide a framework for studying these genotype-phenotype relationships. So when the long path can have the interconnections, and these interactions can be in the form of protein-protein interactions, they can be metabolic networks, transcription regulatory networks, virus host network, and other networks like gene disease network or phenotypic disease networks. So what it provides you, the networks provides you a platform or a framework where you can study the changes which you have observed when you do a case control study, and you can go back to the network and try to understand the biological complexity. So when we say network, basically we are talking about the biophysical interactions, which can be a protein A touches protein B, which is you usually do by yeast-2 hybrid interactions. And you can have DNA protein interactions, you can have non-coding RNA and the target interactions. Not necessarily these are the only interactions. You can also have functional relationships where you can have the activations, repressions, and logic gates, and this also can provide you the links between the two nodes. So there are many repositories which are available, which provides you the information regarding the protein-protein interactions, genetic interactions in different organisms as well as in the human. This is just an example about how many nodes and interactions are known in 2013, but we have gone much far from 2013 to 2017 now. We recently, GEGI has published a Bioplex 2 version in the Nature, where they have more number of interaction coming from co-complexes. Vidal group has published in 2014 an interaction from yeast-2 hybrid, which has 7,000 genes with 40,000 interactions. So we are somehow trying to complete the interactions between the proteins in the human genome. So we have been, this is an example of the interactome which we have been using in our work, which is interconnected and complex. So this is the network which we most of the time use, and it is basically a, you can say, a union of different types of interaction informations, like binary interactions, protein complexes, regulatory, kinase substrate, and signaling. So what we have done is, it's more like an aggregation of the edges. It's not about multi-layer, but is you aggregate all the informations. And the reason behind aggregating was that we wanted to capture as much as interaction informations which we can get to understand the disease. So once you have this hairball which is complex and interconnected, what you learn from it is that it follows the power law. That means you have, this is the power law distribution and the degree distribution of the nodes. And what it tells when you say the power law is that you have a high number of small degree nodes and you have a small number of big hubs. And this corresponds to the robustness of the network, that if you remove the nodes, usually basically you will remove the small degree nodes and nothing will happen to the network. So somebody was talking, I heard yesterday that the small world effect, and that's the property the network has, the biological network has. The second thing is about the distance. So when you look at the distance between the nodes, the maximum distance is 13. That means from one node to another is 13. And the mean degree is 21, as well as the distance from one node you can reach to another node is less than 4. So that means you can reach to from one node to another node in this network by doing a 4 hop, like one to 4 hops. This is the small, I'm just talking about few of the topological property of the network, and the aspects which will be more interesting for this audience is that there is a specific property of the disease genes in the network. And this property is that the disease genes try to interact with the other disease genes rather than other nodes in the network. So if you see the distance expected between these disease genes, it is much closer compared to the randoms, which is random distributions. So this is a property of disease gene, and that's what we have been exploring, that there is the property of the disease gene that they tend to interact more often with other disease gene in the network. So what we wanted to explain here is that the modeling of genotype-phenotype association will definitely require the understanding of the consequence of alteration and multiple scales, and that definitely requires the network where we can model these different types of changes. What we have learned previously is that in 2007, Laszlo built this human disease network, and what we learned from the human disease network was that the two diseases can be connected if they have the common gene. And what happened when they constructed this disease network was that you do find that there is cohesiveness of the diseases which are similar, like different types of cancer and asthma, COPD, or diabetes and obesity. So what we want to focus now is that the topology of the biological network may contain the information about how molecular interactions contribute to biological phenotypes. So what we want to say is that if you explore, like the previous speaker really explored the topology of the network to understand about the disease, you can definitely use, like for example, that the hubness of the nodes is correlated to the lethality, and also the hubness is related to the evolutionary conservation in the biological process. So you can develop the approaches which can lead us to understand the complex diseases. And what it will lead us to, as this is the 5 domains which Eric Green talked about in his Nature paper, that you want to understand the structure of the genome, you want to understand the biology of the genome, you want to understand the biology of disease, and we are in the stage where we want to learn more about advancing the science of medicine as well as improving the effectiveness of the healthcare. So what we believe is that all these 5 components are connected to each other, and there is a network which combine them together to understand the disease. So for example, given that the susceptibility loci for the complex traits are unlikely to be randomly distributed, in the same way the genes which cause the disease are likely to be present in the same pathways, and many of you might have observed this. So if you have a network, and this is I'm showing in the visually, I talked about it previously, that this is the network which we constructed, and this is the visual which contains the biophysical interactions, which contains the regulatory informations. And if you throw so the pink nodes are now the disease genes, And if you throw the disease genes on these networks, what usually happens now is they tend to cluster. So this is the cluster of that particular disease genes. For example, in my case, it is asthma disease genes. And you do observe that they are trying to be more cohesively interacting with each other. So this is the idea of the disease module now, that a disease represents a local perturbations, and you can observe this in the network. We all aware that the current human interactome is incomplete. So if it is complete, you will find all the disease gene connected to each other. But currently what happens is, because it is incomplete, you only find a fraction of the disease modules, and the rest are the edges which need to be discovered. So what you see here in the histogram is that there are very less number of, there are diseases which have high number of genes, but there are very high number of diseases which have very small number of observed modules. This is an example of the multiple sclerosis. You can, as I explained previously, the same network which has edges, which comes from signaling, yeast to hybrid, complex, kinase substrate, regulatory, metabolic, and also literature curated interactions. And when I say literature curated, it is basically a small scale experiments where two proteins have been observed that they interact. That's how people derive these literature curated interactions. There is, of course, there is a biasness for the literature curated interactions, but the reason of currently we are using it is because of the current incompleteness of the interactome. And the sources of the disease genes comes from GWAS and Omium. You do find that there is a small component of the disease genes which are connected, and it is, if you compare this with the random, it is significantly connected. And you can see that they are colored differently here. So this is an example of an observed disease module in the interactome. So till now I was explaining about there is a property of disease gene to agglomerate together. There is an observed disease module. What we have recently observed is that in this disease module there is a property which is the peripherality and the core components. So what till now we have discovered is that there is a positional property. That means when 2007 Laszlo talked about that the disease genes are in the peripherality of the network, which is very much not true now, because once we start getting a complete network, there is a relationship between the high degree and the diseases. But we talk about the positional property. Then the relation property came up in 2015 when we published a science paper where we talked about that there is, you can, with the disease module, you can find out the phenotypic similarity between the disease. And then there is a connection property which I have been repeatedly saying, that the disease genes are more connected together. They don't find a cluster, or they are not part of the topological communities, but they in itself create a disease module. But we try to find whether there is a component property. That means there is two components in a disease module, which can be peripheral as well as the core. And what it reveals when you, this is just an illustration, that if you have a core and peripheral, we assume that the peripheral regions are common among the diseases, whereas core is very specific for disease. Like, for example, autoimmune diseases like asthma, rheumatoid arthritis, celiac, they have common characteristic which is inflammation. That is the peripheral region. And then you have the core, like for example, ORMLD3 is very specific for asthma, and it has been replicated in many GWAS study, but it is not observed in other autoimmune diseases. So what we do to prove our hypothesis is that we go to the gene expression data, we only take the expression data where we have case control informations. I use the same human interactome which I have talked previously, and what we are trying to do now is, if you see on the x-axis, it is the fold change, and on the y-axis, it is the z-score, and the z-score corresponds to the uh, the significance of the connections among the high fold genes in the network compared to the random. So it gives you a z-score, and what you're trying to do here is, as you move from low fold change to higher fold change, you find there are two peaks. That means you find that here you have a high connectivity, and when you move forward in the high fold change, you have another component here where you find a high connectivity. So we explored this property, and we say that you have discovered a bigger module here, but when you move forward in a high fold change region, you find the core of the module, and we call it as an encode property, or peripheral and the core property. And we repeated this in 139 geo expression data for different diseases, and we find the same pattern, that you have a peak here, and then the second peak is where you find the core. So we established that, of course, when we use 139 gene expression data, we established that there are the peripheral and core property, and when we randomize the label of the data, and we look for for the pattern, it is lost. So this clearly indicate that when you randomize the data, or when you see in the random samples, you don't find the same patterns. So in itself, it establishes that there is, of course, the peripheral and core property. I have 18 minutes, so I will be a bit fast now. So then we look at the topological property. The topological property is like degree. The degree is how many edges I have in the network. When I talk about betweenness centrality is that the nodes, what is the fraction of all the node passes to that particular node, explains the betweenness centrality. Closeness centrality is the average length of the shortest part of the node to the all other nodes. So you look at this topological property, and you do find that there is a high betweenness in the core, there is a high degree in the core, and they have a low clustering coefficient. That means when you look at the neighbors, they have more connections to the neighbors. So we do see that there is a difference in the topology between the peripheral and the core. We also find this interesting observation that the modularity of the core is not higher, but it is negative, which means that which means that they don't find this type of clusters, but they are more stretched in a disease module to propagate the signals. And then the biological significance of the core, we do find that they are more disorder protein, they are more drug target, and they are more disease genes. This is an example in asthma, where you find that the core have more drug targets, it has more disease genes, and it has more disorder proteins. So then we tested this observation, whether this is true, that the peripherality is the property where the commonality can be observed, and the core is where you have the specificity of the disease. So we measure the Jacquard coefficient between the peripherals. So here you can see there is very less Jacquard coefficient similarity between the core, whereas in the peripheral you do find there is a very high similarity. So, and you can see that when you calculate the p-value, there is a significantly higher overlap for the peripherals. And this is an example of in the three-dimensional space. You can see that the green one is the common peripheral, which is really cohesive in this cardiomyopathy example, and you can see that the core are really distinct among this disease. So I have 15 minutes. So we also hypothesize that the core passes the signal through their neighbors, and this is an example of autoimmune disease. We tested with an example of vitamin D pathway, and we do find that the vitamin D pathway is less enriched in the core, but it is highly enriched in the first neighbors. That means they transfer the signal for this particular pathway to their first neighbors. So so this is all we learned from the encode idea. I will move forward and talk about the idea of, till now we talked about how disease module is located in the interactome, whether there is the property of peripherality in the core in the module. The next is, can you use the disease module to understand the closeness and the separation between the diseases? So this is a network-based separation measure, which is, in a simple term, it is about the path. So the blue ones is disease A and disease B, and what you are trying to do here is you want to measure or quantify that what is the closeness or overlap among these two diseases. So you do it by the calculating the distance of the genes for disease A and disease B, and as I previously explained that you divide these distances by the diameter, and this measure is called as SAB. But in a simple term, what you can understand here is that if you have a negative separations, that means the diseases are closer, but if you have a positive separations, the disease are far. And we tested this by considering the biological process, molecular function, cellular component, co-expression, symptoms, and comorbidity. And what we observe is that when you have negative separations, you have really high cohesiveness in the biological process. You share the symptoms, these genes are co-expressed, and when you have the positive separations, that means you are really far, and this particular biological property doesn't indicate, it really supports that they are two diseases are separated in the network. So this helps us to explain that how we can study different types of phenotypes in the network. Like for example, you want to study the separations or the overlap between different metabolic syndromes, and this can be a very good example. This can be very good in apply. You can apply that on the metabolic syndrome, and you can even if the diseases doesn't the genes doesn't overlap, you can really find out whether they are separated or overlapping with the different phenotypes. This is an example of asthma and celiac, which have, I think IR1 is the common gene between asthma and celiac disease, and another is IL-18R1. And what we show here is that this is the most enriched pathway among these two diseases, and you can clearly see that the pathway is clearly talking between the two diseases. So this is the immune network for IgA production, and this clearly explains that these two diseases has the negative separations. They are closer to each other. Their pathway really talks. That there is one common pathway, and that common pathway crosstalks between the two disease modules. So I have 11 minutes. We all are aware that the interactome is incomplete, and we only can identify some component of the disease module. So what we want to do, we don't want to wait for the network to complete. We can probably, what we can do is we can have an approach where we can identify potentially new candidate disease genes. That's what we want to do. Like, we want to test whether there are new candidate genes which can explain more about the disease. So we developed this the diamond approach, and it is very simple. This is the illustration of the approach. You start with the known seed genes, which are the red nodes, and then what you do is you find or you agglomerate their neighbors. I will explain in the next slide how you do it. Once you have agglomerated the neighbors, then you test it by using gene expression data, gene ontologies, pathways, and comorbidity. And then you can use this particular disease module to find out new pathways, disease heterogeneity, or new drug targets. So this is an example of asthma here. The node color is the information which we collected from different data sources like GWAS, MASH, OMIUM, pathway, and literature. And the size of the node is based on the degree. You do find that there is an observe module, which is, if you compare with the random, it is significant. The Z-score is highly significant. But you you also observe that there are many genes, disease genes, which are not connected. So what you want to do in this method is you want to agglomerate the neighbors based on their connectivity significance with the disease genes which you have previously identified. And what you are trying to do now is you are expanding your observed disease module into a bigger disease module, and you want to characterize that you will potentially identify new candidate genes there. So this is how it works. You have a neighbor. What you are doing is just calculating an hypergeometric p-value for the connections of yellow node is more toward the red nodes compared to the other nodes in the network. You calculate a p-value, the guy who has the lowest p-value, you add them, and then iteratively you keep on adding the nodes in the network. So you can ask me a question that if you are adding all the nodes in the network, where you want to put a cutoff that this is my disease module. So what we did is we use different data source. Here we use gene expression data of asthma, the specific pathway of asthma, the gene ontology similarity, and the comorbidity analysis. And we test for the significance and we stop where the significance drops. So in this case, it was 350 genes. And it is an NP hard problem. Whenever you apply such approaches, you have to provide some support, and in this case we are providing a support using the different biological data. And then once you have put a cutoff, in this case it was 350, you have a bigger disease module now, and you have potentially new candidate guy for that particular disease. In this case it is asthma. What you can do is you can use this network. You can use gene expression GWAS data to identify the pathways. In our case, we identify these three pathways which were fully embedded in this asthma module, and we were more interested on the GAB1. Another test we did for this particular module was we asked the question whether if I have identified the complete disease module for a particular disease, then if I use any drug response data, then most of the changes will happen inside the module and not outside. And that we are able to establish this phenomena that this is the outside of the module, this is the inside, and when you look at when you throw the differentially expressed genes after the drug treatment, you do find that there is a more clustering of the differentially expressed genes after drug treatment, more in the inside in the module compared to the outside. So this again gives you a platform where you can identify the new drug target. We were able to validate the GAB1 as potentially new disease candidate for asthma. We did the knockout, and what we learned is the glucocorticoid is a asthma drug. In place of glucocorticoid, the GAB1 inhibit NFK kappa B, and there is a reduction when you knock out the GAB1. So in a way, we not only identified the candidate gene, but we also were able to establish that the particular gene is important for asthma. The last topic is previously what I talked about is agglomerations based on the seed genes. But most of the time what happens is you have a particular type of data like genome-wide association, whole genomic sequencing data, where you have the p-values, and what you want to do now is you want to identify the significant genes connection to the moderate p-value genes, and then you want to have a small set of genes which you can try to explore more about explaining the disease. So we developed this confined method, and what it does is you can have a lower p-value to higher p-value genes, and what you're trying to do now is you are trying to add the genes, and you stop where you find the highest connectivity significance among these genes. So you start with the p-value, you apply, you bring the network in, and now you have a small connected component which has GWAS significant and the moderate p-value genes. So this is an example for type 2. This is the GWAS p-value distribution. This is the z-score of the largest connected component, and this is an example of that. Initially you add genome-wide significance, and then next you add the genes which are moderate p-value genes. So in a way, finally what you observe is that you have a connected component which contains the significant gene as well as the moderate p-value, and it can explain more about the pathway and the mechanisms, what is happening for that particular disease. I applied this on asthma and COPD, and the GWAS data. I have 3 GWAS data for asthma and 1 COPD, which has 15,000 cases and 47,000 controls. I use SNP to gene annotation methods like Vegas and Pascal and Magma. These were the conversion of genes, SNPs to gene, and then apply the confine. I stop here for COPD and I stop here for the asthma. What we observe is that once you have identified the module, we look for the overlap. We found that the overlap of asthma and COPD was significant, and also, as I explained earlier, the separations between the 2 diseases was the negative. So what I have identified, the 2 modules, they were again has a negative separations, as well as their average distance was also very less. That indicate that these diseases are close to each other. And once you have this asthma COPD module, you can explore to more about understanding how the 2 diseases are working together. And in our case, what we want to do is now to understand the overlap between asthma and COPD, we overlay different methylation data, expression data on this module, and try to understand how different omics data behave on between these 2 diseases. So I will conclude now that as I have explained you with examples, that network medicine provides you a platform where with the big data emerging from the clinical or electronic health record or exome and GWAS, and we have like big data analytics platform. These all can merge on the network medicine and can provide us the new diagnosis, new therapies, as well as the new treatment for all the complex diseases. And what we believe is that in like 2020, we should think globally and locally paradigm. We know that there are scientific challenges like high quality interactome, protein isoform interactome, complete canonical pathways information is not there, high quality genome annotation is not there. But keeping in mind these scientific challenges, we still believe that the network medicine can take us forward and can provide us the causal network identifications. It can provide us the model to identify the causal genes and path, and it can also provide a model to capture the disease related changes. Finally, I would like to conclude with this, where again network can be a framework on which you can overlay all your different queries and questions. Finally, I would like to thanks the work I did. Some work was done at Barabasi lab, and then I want to thank my group, my postdoc, where like Bingbo did the peripherality and the core, Mohammed did confine, Mark and Arda has helped in other projects. And I would like to thank Ed, Scott for providing the COPD data. And thank you for listening.